Okay, so now we're going to start the actual little lab intro, very brief lab intro. I'm not going to intro the demo, right? That's just how to use Labster. You can do that on your own. But for the biosafety, I already mentioned it's a heavy lab. It's big, 3 hours. And it's going to talk to you. It's not going to really introduce anthrax before it starts asking you questions. So I'm going to tell you a little bit about that. And then you will click on the theory tab, theory. Every time theory pops up in your simulation, make sure to click on it and take screenshots and read it. That's how you get your quiz questions in the simulation. That's how you'll get those correct, is by reading the theory tab. So you'll be, doing everything that pops up, and then suddenly it gives you a quiz and you're like, wait a minute, I have no idea what the answer is. Why do they expect me to know this already? It's because it's at the bottom in the theory tab, and it never requires you to open it and read it. But if you don't, there's no way you're going to answer those questions. Okay. So you have been warned. And a lot of my questions will be from those as well. Okay. So you're going to go, it's going to talk about anthrax and this bioterrorism threat. And they're going to collect a sample, and the goal is to determine if the sample really is anthrax or not. And so in order to work with that sample, you need to work safely with the potential pathogen. And so you're going to learn about biosafety levels, so the different types of labs and the protocols involved with those labs, to make sure you're safe, and also the integrity of your sample, your work. They're going to talk about the buddy system, which is one of the protocols for biosafety level 3, which is what the simulation is. And the labs that you've been in already, like chemistry, general bio, anatomy, those are just BSL 1, biosafety level 1. The microlab, when it was in person, was BSL 2, and then the simulation is BSL 3. Okay. I've actually never even been in a BSL 3. They're not that common because it's a big operation. Okay. They're going to talk about negative pressure system to keep that safe environment, an anteroom, again, also to maintain this safe environment, prevent microbes from getting out into the environment. They're going to talk about safety cabinets to protect you and protect your work. So you work inside of a safety cabinet, which you may or may not have done before, but you'll see it in the simulation. And then the test that you'll run is you'll do a culture of this sample. They won't show you, but they're going to do a DNA isolation. They'll tell you about it, but you won't actually do that part yourself. But you will do a sterility test, and then you send off the isolated DNA for analysis, and then that comes back and they give you the results. So you only really do culture and sterility tests, and you use other people for the DNA stuff. Later on in the class, I'm sure you'll learn how to do work with DNA, but just not in this simulation. And then you'll end with a fumigation and your test results for the analysis. Okay, so this is an overview. So you should be able to have notes on every single one of these bullet points, and be able to explain everything about each of these bullet points. Okay. A little bit of reminders and heads up to use Labster in the best way, and then that'll be it. Okay, Labster, as I mentioned already, is not going to give you any report or summary when you're finished. So it's crucial that you take notes and screenshots as you complete it, and this is why it took 3 hours. It probably would have only taken 2 hours, but I was taking notes and screenshots. And I mentioned also the questions that it's going to ask you during the simulation. You need to use that theory tab if you want to get them correct. And that's how you earn your credit. So Labster might say, like, that it's out of 100 points, or sometimes they'll even show a higher number as it keeps track of your score at the top. But that's going to be converted to be out of 4 points for our class. Okay. And it's automatically done, and it'll be in the grade book on Canvas. You are free to repeat the simulation as much as you like until the due date. Whatever your highest score is what will be kept. Okay. I think that's it. Okay, so a little bit about anthrax. I forgot. This is why I have slides. Anthrax is a disease caused by the bacterium Bacillus anthracis. Okay, and you might have talked about this in lecture, I don't know, but it's rod-shaped and it forms chains, and that's what we're seeing in this picture here. Okay, so strepto and rod-shaped. Okay, and so anthrax with an X represents the disease that is caused by the bacteria Bacillus anthracis, and Bacillus stands for rod-shaped. Okay. Now, anthrax, the disease, you probably are already familiar with, but it causes a very severe respiratory disease, and it definitely can be fatal, which is why this is a known bioterrorist organism, right? You just need to spread the spores. Someone inhales the spores, and then they can become infected. And so you don't spread this person to person, like a person who's sick with anthrax isn't spreading it through the community, only if you come in contact with the spores, and then those are inhaled. When you're infected, you don't produce spores. The spores will germinate in your lung tissue and become regular multiplying bacteria. Okay, so and when that's happening, it releases toxins, which causes the severe respiratory disorder. It makes it very, very hard to breathe. And so typically people will be hospitalized. And in a lot of ways, I guess, kind of similar to what's going on right now with the pandemic, as far as the respiratory disease. Okay, so what else you need to know? So I already told you Bacillus anthracis is the bacteria that causes anthrax. These are rod-shaped bacteria, and they are what we call gram-positive. And that might not make sense to you right now, but it will in a few weeks, okay? But gram-positive are always purple. And if it was gram-negative, it would be pink, okay? And actually, it looks like this little corner here has pink, which just means they started to over decolorize this stain, which is, it's very easy to do. And so whenever you are looking at bacteria under the microscope, you don't pick out the couple of examples that are different. You look at the majority, and if the majority are you know, this dark purple, then it's gram positive. If the majority were the hot pink color, then it would be gram negative. Okay. They're non-motile. They do not have flagella or any other means of moving. Okay, so they're non-motile, but they do form spores, which we talked about. That's how you inhale the spores. That's how you get sick. They have a special coating called a capsule, and this gives them extra protection. So it makes it harder for our immune system to grab on and eat it. And so grabbing on and eating it is the slang way of saying phagocytosis. And as I mentioned, this is a bioterrorism organism. And it's only a bioterrorism if someone has intentionally released the pathogen in order for it to cause disease. And that can be to cattle, which is actually the most likely if in reality, if we were going to have a bioterrorist threat with anthrax, they would probably target cattle, or a different disease to target crops. If you're trying to hurt a civilization, right, a community, a nation, you don't have to just attack the individuals, right? If you take out the food chain, then you've hurt the individuals indirectly, right? So maybe that opens your eyes up a little bit. So that's Bacillus anthracis. Remember, the first time you write out an organism, full name. Then subsequent times you can abbreviate only the first name, and it should always be in italics. Okay. So last bit about Labster, close all of your Windows apps so that the only thing running is Labster. It is heavy, heavy software. It's hard, very hard on your computer. They also probably didn't code it as well as they could have. So when you're loading, you cannot click away onto any other tab. You have to stay on just that tab and wait, maybe go walk down your hallway and back, get a glass of water or something. It will take a few minutes to load. It always warns you it looks like it's frozen at 25, and for me it's always like 23 or 24. So somewhere in the 20s, it's gonna say it's 20 something percent done loading, and then it's going to seem like nothing was happening, and then suddenly boop, it'll be done. Okay, so it's weird, it's frustrating. Just trust us, stay on the tab and wait until it's done. And then when you're doing the simulation, you also don't want to click away onto other tabs, because then you have to reload it and it might not save your progress. Okay, so it's kind of a commitment once you start your Lobster. Now, once you get to a checkpoint, you can stop and reload from that checkpoint. Likewise, if it gets frozen, if it's not giving you the next part of the simulation, you can always go to checkpoints and have it reload to the nearest checkpoint. So you don't have to repeat the entire simulation. And their live hotline is 8 a.m. to 1 p.m. Eastern time. That's 3 hours ahead. So I think 10 a.m. is when they close for us. Yeah. Okay.